morning, everyone. My name is Nathan Spreng. I'm hoping my voice holds up throughout this entire 15 minutes. Please forgive any pips or squeaks or whatever else comes out that's not verbal. If I could have my slides, though. So I am the director of the Laboratory of Brain and Cognition at McGill University. And our lab studies a lot of things, attention, memory, executive function, and how these sort of relate to social cognition. And what I'm going to be presenting today is primarily just one study that was led by a graduate student in my lab, Leticia Muilambwe Chilobo, who presented her poster yesterday. And I'm presenting it because, first of all, it shows kind of a new methodological approach that I've been working on for a number of years. And I'm really happy to share it with you today. And it was just first implemented in a study of loneliness and meaning in life. So it seemed very appropriate for this session today. All right, so loneliness is essentially a discrepancy that occurs between the quantity or the quality of our perceived relationships in life. And really we think this is some sort of biological signal that's trying to attune us to the social ties that may exist in our environment. And the problem is this can have a negative feedback loop wherein we start having hypervigilance to social information and also social threat. And this can lead to biases of memory processing as well as attention towards more negative, salient social information. This is really a widespread public health concern. This is not just an idle thing of feeling sad or lonely. It has direct impacts upon our health. It's related to mortality, risk for depression and anxiety, as well as risk for the progression of Alzheimer's disease. Now, having a sense of meaning in life and a feeling of purpose almost has this inverse relationship. People who have a high sense of purpose tend to express greater social connectedness with their community and confers a broad range of health benefits in addition to just well-being. For example, people who have greater sense of purpose in life recover faster from surgeries. So in this study, what I'm interested in is seeing are these two distinct constructs or are they somehow related, and how is it that they're connected to the brain? There's been some interesting preliminary work looking at the relationship between loneliness and brain structure. In this first study by Kanai, in a pretty well-powered sample, over 120 individuals, showed that self-reported measures of loneliness were correlated with cortical thickness or voxel-based morphometry, one of the two, and showed that people with larger temporal parietal junctions tended to have lower feelings of loneliness. And in fact, this is also controlling for social network size. Regrettably, even though this was a well-powered study, we've had a lot of difficulty replicating this in our lab with equal sample sizes and larger. So who knows? And then there's another study that has shown that loneliness is associated with the underlying white matter integrity of these regions, again associated with the social brain. And then finally, there's been some work looking at meaning in life and its association with cognition and the functional architecture of the brain. I love this study. I think it was really innovative and creative and gave a great treatment for this idea of meaning in life and associated it with, at least in study one, the underlying pattern of connectivity of the medial temporal lobe subsystem of the default network. So these are regions of the brain that are involved in how we simulate an experience, both remembering the past, imagining the future, and imagining the thoughts of other people. So the greater integration within this subsystem of the network, the more self-reported feelings of meaning in life occurred in these participants. So we have some glimpses into the idea that loneliness and meaning might actually be related somehow to the brain, and we're interested in conducting a systematic assessment as to how these self-reported feelings are related to the functional architecture of the brain. So first off, what we see, and we relied upon the Human Connectome Project for our data for this study. We had 942 participants. And just on the behavioral level, you can see that there is a negative association between feelings of loneliness and feelings of meaning and purpose. And there's a number of other covariates that we included in our study, including the Big Five personality, as well as positive affect, and these are all controlled for in subsequent analyses. All right, so we're interested in relating these individual differences in behavior to the functional architecture of the brain. And we take a whole brain connectomics approach. So first off, what we do is, in this case, we had a solid hour of resting state functional connectivity, and then we parcellate the brain we used the 7-network Yo solution and the recent Schaefer atlas having 400 parcels. So we have 400 regions of interest, and then what we did is we individualized each of these parcels, and we fit it to the unique functional architecture of each of our participants' brains. So this is a rather novel feature, so I'm going to go on a bit of a tangent to explain what I mean by this and why I think it's very important. So we know that there's variability in the functional neuroanatomy of the human brain, and that our normalization procedures that rely upon structural landmarks are insufficient to functionally align across individuals. So we see this pretty robustly if you do task localizers, for example. Here's a group localizer of looking at scenes versus tools, but what we can see is that the peak coordinate within each individual subject varies by as much as 3 centimeters in the ventral visual stream. So this is a functional area of the brain that's specific to tool processing, say. And it varies over quite a distance in ventral cortex. Now, and this has been seen in a lot of different studies, but one thing I really like about Dale Stevens' work is that he subsequently shows that you require individual functional localization of a particular point in order to extract the connectivity and then predict behavior. If you rely only upon group seeds, you actually obscure or can't even find or are insensitive to the individual differences or predictions of behavior. And this is problematic, especially when we start using parcellation schemes like the Schaefer atlas and just apply it to a group of individuals, because there's a substantial amount of heterogeneity across individuals. And I think this is really nicely shown by Hishang Liu's group. So Avram had this posted earlier. What we see is that there's a lot of variability in the functional connectivity of the brain, and this variability is present in some networks more than others. So if you're interested in studying visual cortex, motor cortex, fine. Landmark registration is the way to go. But if you're interested in, I don't know, like some of the more juicy bits of cortex in the default network, in the salience network, and the frontoparietal control network, we're going to need another solution because there's high levels of variability. So if you just take a piece of cortex in, say, the frontal lobe that's not very large, this functional region is actually going to move around quite substantially across individuals. So in order to conduct valid between-group comparisons or look at variability across individuals, we need to somehow align them. So fortunately, we have a bit of a solution that we proposed a couple years ago. Here's a toy version of that, and I'll just do a quick walkthrough. What we have, pardon me, is we start off with a number of vertices in the brain and apply a parcel label, and then calculate the functional connectivity across all of these vertices. And then we assess if we shuffle these around and move the parcel designation, can we increase the within parcel homogeneity? In that, can we really resolve that this piece of parcel is a homogeneous area of the brain? And then what we can do is assign a cut point and then relabel our parcel labels. And what we find is that through a number of different iterations, we can approximate to a much greater degree the individual functional architecture. But what's key here is that we're able to also preserve the labels across individuals so that we can then make comparisons in the underlying correlational structure. So there have been other groups that have done an individualized parcellation within subject, in single subjects, and what you see is that, yes, there's variability, and you can approximate the number of regions in each individual subject's brain, but there was not a good way to line them up after the fact. Now, one of the limitations is that we don't actually know how many brain regions there are. In the initialization, we have to start with a common set. Although we find that it's pretty good across any methods of parcellations. So here's a simulation where we have our initialization and a number of different subjects, and we simulate, and after a number of iterations, our error rate goes way down, and our parcel structure can move around from the initialization, and even to the point where it moves fully outside of where the initial parcel was located. And I think this is really key because as I showed you with the functional localization, some of these functional regions can move quite a lot. And then in real data, what we see, and this is using the Yo parcellation, after a number of iterations, the homogeneity of the signal dramatically increases and then stabilizes, and then the connectivity variability starts to go down. So we're able to maximize the homogeneity of the signal within each ROI while preserving the large-scale network architecture and actually reducing the variability of these distributed brain networks and regions. And then just to bring it back full circle, what we see is that when we have done this individualized parcellation, we're able to more accurately capture these differences in the functional activation. So here we have a group parcellation. This is the Glasser atlas. This is the group, and then this is the individualized parcels. And this is when participants are seeing shapes over faces. And what you can see is that the functional activation during this task is more fully captured within the parcel rather than being split up. So this seems to us as though it's working as it should. We're able to capture the functional regions of the brain across our subjects in a way that respects the variability in their functional neuroanatomy. Okay, so we do this. Now we're going back to loneliness and meaning in life. So we've individualized our parcellations, and then it's kind of what we do. We extract the time series from each of these regions. We calculate our functional connectivity matrix, which is essentially a correlation, and then we do an R to Z transform. Now with 400 regions of interest and looking at the interactions between all of them, what we have is 79,800 unique ROI to ROI correlations. We're pretty much back to where we were before in voxel space. We have a lot of things to look at. And Avram is doing Bonferroni correction, which is misguided. And what we really should be doing is a multivariate approach that actually recognizes the fact that there's a covariance in the architecture underlying whatever associations we might find between brain connectivity and behavior or patient groups. So canonical correlation analysis is one approach. I use partial least squares. And here, basically, the underlying math is quite similar. It's a singular value decomposition, which looks to have the maximum linear association between a variable of interest, be it group or loneliness, and then relate it to that connectivity. We then use permutation tests to assess the significance of the relationship, and then bootstrap sampling to assess the reliability. And from our multivariate output, we can see how we have behaviors related to the connectivity, and then the underlying matrix that expresses the patterns of pairwise associations that are related to those behaviors, as well as the amount of covariance explained. And then we can do post hoc tests leveraging graph theory to assess, what are the aspects of this network that are meaningfully related to behavior, so that we can start to interpret what this matrix actually means. Okay, so when we do this, looking at loneliness and meaning in life, what we find is that across the entire functional connectome, there are a number of regions of the brain that show both positive and negative associations with these behaviors. So what we see is that there is, a modest correlation between meaning in life that's positive with this matrix and a negative correlation with loneliness. So what that says is that the higher levels of loneliness people have and a lower level of meaning, that's associated with greater connectivity in the blue regions. However, for people who have lower levels of loneliness and a greater sense of meaning and purpose, there's greater positive connectivity between these regions. So it's harder to see the gold or the warm colors. There's not as many of them, but they're in there. And I'll show you that in just a moment. So what we see, again, is a whole brain connectome and its association with loneliness and meaning. So what we can do, as I mentioned, is take some graph analytic measurements, and these are approaches to look at modularity or centrality, the extent to which regions and networks talk to each other or are more insular. So we use the measure of segregation, which is an assessment of within versus between network connectivity in order to assess this network. So because we know the labels of these networks and how they should be instantiated, we can assess whether or not there's a prevalence of connections within the system or a prevalence between these systems. And in fact, this is where some of the strongest differences really emerge, and we can start to interpret this underlying matrix. So what we see for people who have high levels of loneliness, there's a disproportionate number of connections between the frontoparietal control network and default network with the visual and the somatomotor network, as well as the ventral attention network or salience network. So there's a preponderance of between network connectivity that's largely driven by the frontoparietal control network and default network. Now, in contrast, people who have higher levels of meaning in life and lower levels of loneliness tend to have more within-network connectivity across systems. So the networks that are on the diagonal have more positive connections. So we see the dorsal tensions connected with itself, the ventral tension with itself, the limbic, as well as the default network. In addition, we have the default network. There's greater between-network connectivity with the frontoparietal control in the limbic network. So by and large, there's more modularity of the network systems and people who have greater sense of meaning in life. However, there seems to be some important connections as well as across networks. Then finally, it seems like the default network might be one of these key networks in terms of higher levels of meaning and lower loneliness, this connectivity with the frontoparietal and limbic, versus default network connectivity with the visual and somatomotor network. So with that, I'm just going to wrap it up. I'm just going to say that meaning in life and loneliness are clearly negatively associated. That might be reflecting this underlying mechanism. They're both associated with distributed patterns of resting state functional connectivity. For negative aspects such as loneliness, it's more associated with between network connectivity versus meaning in life with more within, and that network neuroscience methods can flexibly integrate whole brain connectomics, the analysis of individual differences. So with that, I'd like to acknowledge again Leticia Muilambo Chilobo. This work just came out in SCAN. I think it should be in the April issue. And then there's a number of people involved in this project, our funders, and thank you very much for listening.